Hello grade 12s and welcome to Life Science, and today we're going to have a look at the code of life, right, which is very difficult because codes and understanding them is strange in my head, right, but this code is actually quite cool. Let's have a look at it. We always start with terminology, right, and the few words that I have for you, there's 4 of them. First one, I want you to tell me what the meaning of the following words are: DNA, RNA, meiosis, and mitosis. Those 2 words that always come up and we always forget which is which. Right, I'm going to give you two minutes to quickly do it. Are you ready? Off you go. Well, let's have a look at these meanings. Okay, first one, DNA, right? We always knew it as giving you this is why you look like your mom or dad, because of your DNA and it's true. So let's have a look at what it really is. DNA stands for deoxyribose nucleic acid, right? And exactly what you said, it carries your genetic information. Okay, now, strange enough as it is, the reason why it carries your genetic information, because it makes proteins. And the proteins are what makes you look like you do. Right. Next one, we have RNA. RNA is very, very similar. Okay. Well, in the name-wise, the only thing it doesn't have, if we have a look at it, deoxyribose nucleic acid, and RNA is ribose nucleic acid. Okay. It doesn't have the deoxy in it. Okay, and it acts as a messenger to carry information from DNA for controlling the synthesis of proteins. Right, that's all it is. It is the carrier of information from one place to the big factory that makes proteins. Okay, now we've got these 2 words that we always, well, a lot of us get confused about. First one, meiosis. Well, let me tell you both, meiosis and mitosis. Okay, meiosis is very similar. It's cell division that results in 4 daughter cells, each having half the amount of chromosomes of the parent cell. And we use meiosis, and there's only one place it's going to happen in the body. Okay, it's where you make your sex cells. In other words, sperms or eggs, or should I say the gametes. Okay, the male gametes and the female gametes. When it comes to mitosis, remember the T in mitosis tells you 2, instead of meiosis, which gives you 4. Mitosis, it's cell division, okay, that results in 2 cells, each having the same number and kind of chromosomes as the parent cell. Remember, mitosis gives you identical daughter cells, where meiosis gives you unidentical cells. Okay, so those are the 2 and the difference. The T in mitosis tells you only 2 cells. Okay, it's the best way to remember it. Now, let's have a look at this DNA things, as we were talking about in the beginning. Okay, DNA, its real name is deoxyribose nucleic acid. So it's a nucleic acid with oxygen, or deoxys, right? Now it's found in the nucleus as genes and chromosomes. So you got your cell, right? It's got that cell membrane, and inside you get the nucleus, and inside the nucleus you'll find your DNA, all the information that you need. This is going back to long time ago when you were in grade 8 to 9, just to remind you. Okay, it's found outside the nucleus as well. Okay, so you get your DNA inside your nucleus, and you get outside your nucleus as well, and that is called your mitochondrial DNA. Now, your mitochondrial DNA only comes from your mother. Okay, not from your dad, only from your mother. Okay, now let's have a look at what DNA consists of. Okay, this is a beautiful picture of DNA. It messes it up and it turns it around and everything. First of all, let's have a look at right here at the bottom at this red and green like ladder thingy that's twisted. Okay, first of all, it's double, which means you'll hear it. It's called a double, and then it's twisted helix. A helix means that you've taken something and you've twisted it to go in a funny shape. Okay, that means it's a helix. Okay, so this is called a double helix because it's two of them that's turning around. Okay, so that is your double helix. Okay, then you will notice on the left hand side over here, we've unturned that double helix and we ended off with two strands, the green and the red. Okay, and the green and the red, they've made up of certain things. Okay, so let's have a look at the nucleotides itself. Okay, and there they are. Let's have a look at it. Okay, now you will see very clearly that it's made up of three things. It's made up of a P, it's made up of a D, and it's made up of any type of letter, which is T, A, C, and G. Can you see them? Those are the three the three things that one nucleotide is made up of. Just a quick cap. Okay, first of all, the P stands for phosphate. The D is a deoxyribose pentose, meaning 5, sugar. It's a sugar molecule. And then the last one, you get this piece over here, if you see me circling them, okay, those pieces over there, which are called your nitrogenous bases, okay, and you get 4 different types. Don't forget them. You got G, which is guanine, T, thymine, A, adenine, and C, cytosine. Okay, now you're going to get these big words as well. They are called purines and pyrimidines. Okay, the way I remember it, purines and pyrimidines is big. They work opposite. When I look at pyrimidines, I look at a single, if you have a look at it, it looks like this. One of those, okay, is a pyrimidine. When I get to a purine, it's got this shape. Okay, it's got a helper, there's one of them, and then it's got the second part which comes out like that. See, it's a double. A purine is small, a pyrimidine is a purine is big, and a pyrimidine is very small. If you have a look at your purines, so the big ones, there's 2 of them. You get the adenine and guanine, and then of course the cytosine and thymine always go with it. Okay, so they are the pyrimidines and purines. And then another thing that everybody gets confused with, if you have a look at C and G, they're a common shape. If you have a look how I drew it there, all I'm missing between the C and the G is a little line over there. Okay, so your C, there they are, the G comes up and makes a little line. So those 2 fit together, and then of course A and T fit together. Cool, huh? Those are just purines, pyrimidines, guanines, cytosine, thymine, and adenine. Double helix, and a double helix forms a the DNA molecule, which altogether, okay, is what makes you who you are. Cool, Hab? Now, that was an overall. The role of a DNA. Roles of DNA. Simple, replication, making new cells. They've got to go through cellular making new ones. So in other words, let's say mitosis, okay, and then you get your DNA replication, making new DNA molecules. You've got encoding proteins, in other words, making proteins, certain types of proteins, okay. You get cellular management. In other words, it tells the cell what to do, okay. Then you get the ability to mutate, in other words, change, become different, okay. Mutate means sometimes better, sometimes worse, okay. A little mutation, let me tell you, albinoism. So if I have a look at the king of the jungle, the lion, right? You get the normal color lion, and then you get the albino, so the white lion, okay? It's a mutation, okay? Which means it's not always good or bad, because the white lions don't last as long because they don't camouflage as well. But then, of course, you get the mutation like Spider-Man, which is a cool mutation, which I like that mutation more. They aren't really true. Okay, and then the last one, replication, cell cycle. You need to look at the replication, which has got to do with your cell cycle, which you learnt in grade 10, right? And it's necessary. You need to understand your mitosis and your meiosis and all of that. So don't forget them. They're very, very important. Okay. Now, let's have a look at DNA replication. This is the process, okay, of mitosis, tosis, okay. You look at it, you remember you get your interphase where the cell grows up and becomes normal, okay, normal size. Then you get to your different cycles, which you need to know off by heart. So you get your prophase, metaphase, anaphase, telophase, okay. Your prophase is where they start linking up with their pairs. Your metaphase is where they start lining up on the equator. Your anaphase is where they're starting to be pulled around from your to the different poles, and then the telophase is where you get cytokinesis, which makes two different cells. So that's how it works. Okay, remember a nice one of looking at it as well. I'm just going to extend this page quickly so I can show you. Okay, your DNA also replicates. Remember, it can split. So I'm just going to do lines like this. Okay, they're going to split to make two new DNA molecules. Your other DNA molecule is going to come and fit in there. Okay, and you're going to make two brand new DNA molecules from this, which means you can make another cell. Can you see how I've made your DNA? And as you split further and further, I'm going to erase this so you can see. Let's do that. Oh, what happened there? Let's undo. One more time. There we go. Okay, it's going to come down here. This one will come over here, creating new DNA there. And this one will come here, making new DNA there. So you've got two new DNA strands. That's how DNA replication actually happens. Okay, so you get a chemical that makes it do that, which is DNA polymerase, which I don't think you really need to know that badly, but it's nice. The DNA polymerase is the ones that split the DNA molecule open. Okay, so it splits it and it puts it back together. Okay, so that's a nice way of looking at it. This is quick. Remember, it's very important to know your textbook. Get to understand this. If you understand, it becomes very, very easy. Okay, now here we go. RNA. RNA is a big thing, okay? RNA, you get different types, and that's why they're so important. So I'm going to run through what RNA you get, why they're there, and then we're going to start looking at how we actually go through this process of making RNA or making DNA or making of proteins, which is very important. Okay, let's have a look. RNA as it is, okay? It stands for ribose nucleic acid. Okay, there is different types. Okay, if we have a look, there's mRNA, which is your messenger RNA. And your messenger RNA is exactly that. It just carries a message. So it transcribes, very, very important word to remember. It transcribes the genetic code from DNA into a form that can be read and used to make proteins. That's the biggest thing about RNA. Okay, the mRNA carries genetic information from the nucleus to the cytoplasm of the cell and all the way to that protein factory called the ribosome. Okay, because a ribosome is the how do you say it? The be all and end all. There we go. The be all and end all of making proteins. Okay, so that's the first one we're looking at, mRNA. I haven't told you much about it. All you know is it goes through translation. It goes from the cell membrane, gets the code from the DNA, and goes to the ribosome, which is in the cytoplasm. Okay. Next one that we have your rRNA. Okay, rRNA is ribosomal RNA. That's why. And remember, guys, it's small m, RNA. Okay. If you put a big M, it's wrong. Okay, so it's a small RNA, and the same here, small r, and then RNA. Okay, and what it does is it's located in the cytoplasm of the cell where ribosomes are found. The rRNA directs the translation of mRNA into proteins. That's what it does. And then I've got one more for you. Okay, one more if we can get there. There we go. One more, which is your tRNA. Okay, and it's called your transfer RNA. Make sure you remember it's a small t. tRNA, which is located in the cytoplasm as well, and is involved in protein synthesis. Okay, it brings and transfers amino acids to the ribosomes that correspond with each other. So certain ones go with certain things. Okay, and then it's very important to remember that each nucleotide has a codon of an RNA. Okay, so we'll talk about codons and anticodons in a few minutes. Don't stress about that. Next thing, you have the amino acid that can be joined together to process a polypeptide or a protein. In other words, we're making a big protein. Okay, polypeptide, or you get a dipeptide, which is only two amino acids making one protein. Okay, so that was a lot of information, definitely. So I think you need a break, because I do, and I will see you straight afterwards. Cheers. Okay, welcome back. Now, I've had a look at the next slide, and I've come to show you the different things, because you need to be able to identify the difference in RNAs. Okay, now we all know that RNA and DNA, what they're actually used for, because we've done it before. Remember, this is just something that I'm trying to bring to life and remind you of it. Okay, so first of all, messenger RNA. It's only one strand. All RNA is only one strand. Okay, second of all, you will notice that they are different. Okay, you'll be able to identify them immediately. Okay, they are completely different to each other. Have a look at them. Okay, now one thing that makes RNA, one of the biggest things that make RNA different to DNA, okay, first of all, single strand, but DNA can split and make single strands, am I right? Just for a few minutes, but it can split. So there could be a time where you take a photo and it's just at the wrong time when they open. Okay, so let's say that's not a big thing. The biggest thing is, remember we spoke about this A, T, G, G and C, adenine, thymine, guanine, and cytosine, all of those guys, right? Remember we spoke about them. Okay, now in a DNA molecule, you will find all of these. Now remember I said A goes with T, T goes with A, G goes with C, and C goes with G. So we've got them. Now in an RNA molecule, remember there's a difference between RNA and DNA. One is a ribose nucleic acid, the other one's a deoxyribose nucleic acid. Okay, which means when it comes to the ribose nucleic acid, okay, it is the best way to remind yourself is it's not a very strong, because you know it's single strand. Okay, so this is how I remember. It's not actually true, but this is how I remember it. Okay, it's not really strong, so it cannot carry T. It cannot carry a ton. Okay, so it cannot carry this T. Okay, so instead of getting a T, it carries a U, a unit, which is uracil. Okay, so if I had a look at it, DNA has got these ones. DNA is ATGC. RNA is AUGC. Can you see the difference? That U, as soon as you see a uracil, you will know it's a DNA. It's a RNA molecule. Okay, that's the first thing. Next thing, the two that's very different. It's ribosomal RNA is not actually that different to messenger RNA. The one that you got to have a look at is this tRNA. Have a look at it. It's got this funny structure up here. Okay, it's got a funny structure up there. Then that little structure is very important because it's holding, okay, it's holding an amino acid, and each transfer RNA or tRNA is a specific one. Okay, you get them as an anticodon. Okay, and they got three. They're made up of 3 anticodons or 3 nucleotides. Okay, and each 3 holds a specific protein, a specific amino acid. So let's see, adenine, adenine, adenine holds a specific protein. A different one would be adenine, adenine, uracil will hold a different amino acid. If it was adenine, adenine, guanine, a different amino acid. Okay, so watch this. Different shape between tRNA and rRNA, okay, and mRNA. tRNA has only got an anticodon, so it's by itself. If I have a look at messenger RNA and ribosomal RNA, it's one long strand. You see, there's 3, then there's 3, then there's 3. We've cut off this by accident. Okay, so there's 3 in each. These are called codons, but the codons are joined together, whereas the tRNA, it's not. Okay, so that's just showing you exactly what the mRNA, the rRNA, and the tRNA actually look like. And remember, uracil, very, very important because it's only with a RNA molecule, no DNA. Okay, now, where does this messenger RNA, we'll talk about messenger RNA in a few seconds, but where does it actually get, where does it actually come from? Where does it go to? What does it actually do? Okay, so I've got a couple of questions which I want you to go through, okay? And I think I'll explain how things work in a few minutes, straight after this. And so let's do this question first and see how you do with it. Okay, the diagram below shows part of a DNA molecule. Okay, now I want you to have a look, and it says there, you've got all these different parts, okay? And the question is, the correct label for part X, Y, and Z represented are. Okay, first one. Let me just see if I can bring this down a little bit more. How many are there? There's D. Okay, so let's see. I'm going to give you 2 minutes. No, I'm going to give you a minute. Half a minute, like that, and then half a minute I'm going to bring it down. Okay, so make sure, in actual fact, let me do this. I'm going to put a Z over there to make it easier for you. I'm going to put a Y over there to make it easier for you, and an X over there to make it easier for you. So you can see all of them. How cool is that? And I'm going to give you a minute to tell me exactly which is which. Okay, are you ready? Off you go. Hello. Now, if we have a look at this, it's very simple. Okay. In the beginning, I told you exactly which goes where. So let's have a look. A, is it deoxyribose sugar? Okay, we need to go into the order first of all. X, Y, Z. So we know that this one must be X, must come first. Okay, so that one, if I remember carefully, it was something like that with a P in it. Can you remember? Okay, it's very important to remember that. That's a phosphate. So Deoxyribose, is that it? No ways. Okay. Is it a ribose sugar? No. We're looking at the X. So it can only be this one or this one. Okay, the phosphates. So it's either it's either B or D. C is out of the question, definitely. A is out of the question, definitely. Okay, then we need to have a look. It says ribose sugar, okay, and it says, let's see, deoxyribose sugar, okay, and then lastly, it's got weak hydrogen bond or a hydrogen bond. Okay, now, a lot of places forget about the weak hydrogen bond. It has to be a weak hydrogen bond. If they just put their hydrogen bond, they're going to accept it. But I'm telling you now, it's a weak hydrogen bond. Okay, it has to be able to split and come together very quickly. Okay, please don't forget that. Next thing, it cannot be a ribosomal sugar. Okay, it has to be a deoxyribose sugar because remember, look here, it's a pentose sugar. You see, it's got 5 sides. There it is. Okay, and it's got another 5 sides over here, which means both are pentose sugar. If there's a double together and the sugars are identical, it means it's deoxyribose. That gives you the answer of B. So B is the correct answer and D is the wrong answer. Okay, please don't forget about the weak hydrogen bond, very, very important. Okay, now, this is the process of protein synthesis. Okay, and I've muddled them all up like you have no idea. Okay, and this is the coolest thing in the world. Well, in my eyes, it's the coolest thing in the world. Okay, I want you to have a look at it. This white barrier is your cell. Okay, it's your cell membrane. Okay, this green barrier over here, right, that is your nucleus. And what do we find in the nucleus? You find your DNA strand, which is represented over here. Okay, your DNA strand. Now, what happens is we've decided that we need more protein, we need more muscle, for example. Okay, so we need more muscle. So the DNA says, okay, we need more muscle. So we need to make some protein. So the DNA that makes muscle has to split and open. So it calls along a thing called a RNA polymerase. It's a little machine that unzips. It's like the zipper. Right. So it calls this thing called RNA polymerase, and it starts working, and it unzips this DNA. And as it unzips, you have an RNA molecule fitting in there to get the right code. So every time, so if I have a look at it, this DNA molecule that's split over here, okay, there it is, 1, 2, 3, 1, 2, 3. I'm going to do it like that, 1, 2, 3. So every time it splits open a bit, an RNA molecule comes along and sits there, and comes along and sits there. Okay, now as that's happening, you'll notice it unzips, okay, so it unzips like this, so it stands there, it unzips, and then it unzips and it closes at the top there, and it unzips and lets it go. So what actually happens is it creates a wave-like structure. So it unzips and then comes back together. Every time it unzips, the right code comes and joins on messenger RNA, leaves, and then the DNA comes back together. So now it's got this code. So this RNA molecule, okay, the mRNA molecule has got this code. It knows which protein to get. So it leaves the nucleus and it goes into the cytoplasm and it reaches that huge protein factory. Right, that protein factory, what does it do? It invites it in and it starts saying, okay, all the tRNAs, the transfer RNAs, come along, let's see if you fit. So here you've got this, remember, DNA's got a triplet, okay, they're in 3s. Your mRNA's got your codons, and your tRNA is your anticodon, okay? So you get this mRNA and it's sitting there, and there's quite a few of them. So let's say there's like 50 or whatever it is, right? The tRNA comes along and goes, do I fit here? No, wait, I must wait my turn. And the next one comes, oh wait, I fit there. And as it does that, it releases the what do you call it? The amino acid, and leaves, and the next one comes and fits next to it. I fit here, and the two amino acids, they join together through a process called dehydration synthesis. So they lose water and they join, they become a protein. Okay, so that is that whole protein, or that is that whole system that's showing over here. So let's have a look. DNA molecule, there it is. RNA is taking, mRNA is taking the message. Here's the RNA molecule again. The tRNAs are coming along with that amino acid. Can you see it? The amino acids join together to form a protein. That's quite cool, isn't it? I also thought so. Now, little question for you. Let's see how good you are. Okay, you've got to study the diagram below and answer the question. This process is on protein synthesis. Okay, so here we go. It says, identify the stage of protein synthesis that is shown in the diagram above. Okay, I want a little bit of explanation with it. You must be able to understand and know why it is. So I'm going to give you a quick minute to do it. I'm going to give you 2 minutes to do it. I want to know what process it is and why you say that. Okay, are you ready? Off you go. Let's look how you did. Now, your question was, what process is this? And this is translation. This is the process called translation. How do I know that? First of all, we've got a single strand here, which means it's not DNA. It has to be RNA. Okay? And then the second part, you've got these little things over here, which is going to be your protein, and you're going to have these little ones floating and bringing along. You can see they labeled amino acids. They are your tRNA, and they're going to come along and join the rRNA, which is in the ribosome. Cool, hey? That was good. Now, a little bit of break for you quickly. When we get back, I've got some more questions for you. I'll see you straight afterwards. Cheers. Welcome back. Now, some more questions. The more questions, the better it is. Okay, so same thing, same diagram, and the question was, identify molecule X and Y. And I'm going to give you a quick minute. Remember, I always want an explanation. 1 minute, are you ready? Off you go. So let's have a look at these things. First of all, okay, what is, let me bring it down slowly, I don't want to give you the answer yet. Identify X and Y. So we're looking at X and we're looking at Y. You know it automatically. It is your X is your tRNA and Y is your rRNA. Why do you say that? Okay, first of all, X, if you have a look at it, it has got anticodons. They are in 3. They are not joined. Yes, there's a line giving out, but that's just a label. Okay, it looks simply all by itself. There's one, and it's in 3s. So we know that's a tRNA because tRNAs are found in 3, and it looks different because it's got this little molecule that is holding the protein or the amino acid to it. Okay, we know that Y is your rRNA. We said rRNA. We know it's your rRNA. First of all, it's found in your ribosomes, but that's not good enough because so is your tRNA at times, right? But it is a long strand, a very, very long strand. Remember, rRNA can be lots of codons joined together. And we know it's a RNA molecule because of the U. Okay, because of the U, it can tell you exactly it's a RNA molecule, and an RNA molecule that is found in the ribosome is ribosomal RNA, and the RNA found outside is your mRNA. Cool, that was simple. I know you guys have gotten that. Very, very easy. Next one, same diagram, and it is state the term for the group of 3 nitrogenous bases indicated by V. Okay, what is the 3 nitrogenous bases stated there? And you've got a minute. Please, can you do me a favor before you take that minute? Make sure you give me that and the other 2. Let's see if you can remember them while you're at it. Okay, but mainly for V. Are you ready? Off you go. Let's have a look what we've got. Now, V is very easy. What is it? It is an anticodon. Anticodon means opposite to a codon. Okay? Can you remember what mRNA and what rRNA have got? Can you remember? Okay? They just have codons. There they are. They just have your codons. And then lastly, what is your DNA? The 3 in a DNA, what are they called? 3, it's like triplets. That's all it is. It's like having triplets, right? That's why we have 3 triplets. So they are your triplets. Your triplets join to your codons. Then your codons go off and join to your anticodons. Can you remember those? Very important that you remember those words. They are the ones that are very important lately. Next one, same slide. Okay, give the nitrogenous base on the DNA strand that codes, look at this, give the nitrogenous base for the DNA strand that codes for UAU on molecule Y. Okay, where's Y? So there's Y. You need to give the DNA base that codes with that. And I'm going to give you a minute. Make sure that you give me an explanation. Don't forget to give me that, right? Are you ready? Off you go. Okay, before I carry on with this, please remember you have to read your questions carefully because they will confuse you. Okay, and it's one of the questions that people do forget. First of all, if I read it, the first thing I saw was this big word here, DNA, deoxyribose nucleic acid. Okay, that's the first thing I saw. That means there's going to be no such thing as U's. Okay, so they said there, give the nitrogenous base on the DNA strand that codes for the base UAU. Okay, now, can you remember? You have A, you have T, you have G, and you have C. What goes with C? G. What goes with G? C. What goes with T? A. What goes with A? T. Remember, those are the ones that join together. If it comes down to RNA molecule, you don't get that. So let's say this is the RNA, and this is the DNA. Okay, remember there's no such thing as T, we've just got a U. Okay, now they're asking for UAU, so let's have a look at it. UAU is going to be TAT. So let's see if we can get that right. So let's see. UAU, which one goes with U? It is A. Okay, which one goes with A? T. Which one goes with U? A. So the answer, there it is. Okay, remember you can have T in DNA. So the answer is ATA. They asked for the one on the DNA strand. Very, very important to read your questions carefully. Right, okay, here we go. Another one. Okay, and the question is, name and describe the process that occurs in the nucleus that produces molecule Y. What happens to produce that one in the nucleus? Now this is quite a big one. So I'm going to give you 3 minutes to do this, okay? Concentrate, give me everything. Okay, are you ready? Off you go. Let's have a look at this process. Remember, it was a nice long process, so we know exactly what it is. I've put it down here in point forms. First of all, the process that we're looking at is called transcription. Okay. Now, a double strand of DNA molecule unwinds, so it unzips. Okay, remember, it unwinds and unzips. Then the hydrogen bond breaks. That is why it's got to be a weak hydrogen bond. Okay. One strand is used as a template for the mRNA molecule to get the code. Right. Next one, using free nucleotides from the nucleotide pool, which is also inside the nucleus, right? The which is found inside the nuclear plasm, right? As I said, you're using your nucleotide from there because there's a huge pool of them. Next one, the mRNA is complementary to the DNA. In other words, A goes with U, T goes with A, C goes with G, G goes with C. And that's what we're trying to say here. Okay, and then lastly, this process is controlled by an enzyme. Right, the enzyme does it. This enzyme that takes place, can you remember what enzyme it is? Because if we're working only with DNA, it's called DNA polymerase. If we are making protein, if we are making proteins and we're going to use RNA molecules, it's got to be RNA polymerase. Cool, hey? So hopefully you got all that right. Now, before we finish this off, this whole section, I just want to go one thing through you. Please don't forget, okay, when we're talking about meiosis and mitosis, it was something that I remembered right in the beginning. Remember, meiosis and mitosis. Meiosis have got two stages, meiosis 1 and meiosis 2. Okay, and I put this in there for a reason. Okay, don't forget, there's only one interphase, which is not really there. Meiosis cannot, it only happens once. Okay, it can't happen like mitosis over and over again. So you get meiosis stage 1, right? And in that stage, you're going to get a thing called crossing over. Okay, that happens in stage 1, and then you get to your stage 2, where you're going to have a thing called they're going to line up on the equator in different orders, and all of that helps with DNA and changing of DNA and making sure that we don't look identical. So this is the main thing, what we need to look different, genetic variation. Right, that's all we have for today. I'm sure we'll see each other soon, and until then, cheers.